Good morning, everyone. My name's Aman, and I'm the GCSE Chemistry tutor for the MyTutor online school. Today's session we'll be looking at natural polymers and DNA. I know the schedule might have said introduction to electrolysis, but because of problems that we had on Monday, that will now be taking place tomorrow. And the second lesson on electrolysis will be happening next Wednesday. So as I said, natural polymers and DNA today as a continuation of the polymers that we covered yesterday. This is a 1 hour session, so it will last from 9:45 until 10:45. And as usual, if you have any questions at all, please pop them into the chat box or the Q&A function, and I will try and answer them as we're going along. If you haven't met me before, my name's Aman, and I study medicine at King's College London, and I've done lots of lessons with MyTutor. So we get started then. So the learning objectives today, we need to know how sugar molecules polymerize, know how amino acids polymerize, which is a higher tier topic, and understand and explain the structure of DNA. So it's quite biological today. You may have covered this in your biology sessions already, but I'm going to be mainly focusing on what you need to know for chemistry GCSE. All of the exam boards have these topics, but it does seem to be one of the newer topics. So there aren't many exam questions on it at the moment. I've managed to find some that we'll have a go at the end, but the amount of knowledge you need to know about these topics isn't too much, and it's pretty much kind of transferable from what we learned yesterday. But before we go into all of these learning objectives, yesterday we covered condensation polymerization, which is a higher tier topic. And as you remember, I wasn't able to draw the diagram for you, so I'm going to do it today. So before I get started then, does anyone remember how we do condensation polymerization to form a polyester? Does anyone remember the monomers to form a polyester? So think about how we make an ester, what's used to make an ester, and then can you remember what the difference is to make a polyester? Does anyone remember the monomers? So it's the same monomers as making an ester, slight difference though. Excellent, yep, so lots of people are saying diols, good. So diols are alcohols with two OH groups, excellent. So that's one of the monomers, and the other monomer, which someone has said, is a dicarboxylic acid. Excellent, very well done everyone. So I'm just going to draw it out for you today. So if we start off with a diol then, let's think about ethane diol. So ethane has two carbon atoms. So if I draw my carbon atoms like so, and then it's going to have an OH group on one end and an OH group on the other end. So when I've drawn alcohols in the past, we've only drawn it with one alcohol group. So for example, ethanol would just have a H here. It wouldn't have this OH, but this is ethane diol. So ethane diol. And the di just means two, and there's two alcohol groups. So that's the first monomer. And the second monomer is a dicarboxylic acid, which everyone correctly said. So the traditional one they look at is hexane dioic acid. So hexane dioic acid. So that will have 6 carbons. So if I just draw the middle ones first. And what you remember hopefully with carboxylic acids is that a carbon is involved in the functional group. So does anyone remember, before I draw it, what the carboxylic acid functional group is? This is higher, so condensation polymerization is a higher tier topic. So does anyone remember what's the carboxylic acid functional group? So for example, alcohols are OH. What's the carboxylic acid functional group? Excellent, COOH, yeah. So when we do hexane dioic acid, we've got 4 carbons with just hydrogens, but the 5th and 6th carbon are the dioic acid. So we've got the COOH, so that's 1, and then on this side, COOH. So that's our hexane dioic acid. And what happens is these join up to form a polyester. Now in the exam, you wouldn't be asked to draw out ethane diol or hexane dioic acid. In fact, if you are asked to draw it, you can do something very simple, and all you need to do is a square to represent the middle part of the molecule or the monomer, and an OH either side. So this would be our diol. And for the carboxylic acid, again, a square with a COOH each side. So in the exam, you don't need to draw out all the carbons and the hydrogens. You can just draw it with a square and the 2 functional groups. What is a dioic acid? So a dioic acid is a carboxylic acid with two COOH groups. So remember the carboxylic acids, what makes them a carboxylic acid is the COOH, so this double bond O and then the alcohol group as well. So you've got a carboxylic acid there. But in a dioic acid, they have two of them, so one on each end, and that's what makes it a dioic acid. So that's how you would draw it in the exam in the light blue, where you can see this little box here and then the functional groups. And then to draw the ester, I want you to imagine that we've got an alcohol here that we join the carboxylic acid to. So what we'd end up with is the OH group, our square, and then instead of this OH group, because it's joined to the carboxylic acid, remember what's the ester bond? Does anyone remember from last week? What is the ester bond? So COOH is carboxylic acid, OH is alcohol. What's the ester functional group? Does anyone remember? What's the ester functional group? Good, COO. So what I'm going to do is I'm just going to write this a bit differently just to show you how it looks. So I'm just going to write this like this. Okay, just so it's reflected as it would be. So what happens is this OH group has come from here. We've then got the square, and then the OH group here reacts with this COOH. And if you remember from when we formed esters last week, the COO group comes from the carboxylic acid. So it's this COO that comes first. So what we end up having is a COO group that will look like that. Then what's left is we've got this alcohol group and this hydrogen, which is the condensation polymerization because water's removed. So remember in condensation polymerization, we remove water. So if when we make this ester bond, this OH is not there and that hydrogen there is not there, that makes water. So water's removed. Then if I add in another alcohol, what happens next is we're still carrying on with this carboxylic acid. So we started off with this alcohol, we added the carboxylic acid to form this COO group. Next, we're going to have this square, so this carboxylic acid. And then again, we're going to add an alcohol group to this carboxylic acid functional group. So this is COO. And then again, this hydrogen comes off and the OH from an alcohol group. Then we have another square and it's going to repeat. So essentially what I've drawn for you there is what's going to happen when we make our ester or our polyester. So let me just move that along. So what happens here now is we'll end up with an OOC, so OOC, and then next it would be COO, like so. Sorry, then next a square, then COO. So what's happening is hopefully you can see that there's a repeated unit just here. So that's the repeated unit for a polyester. Has anyone got any questions about that at all? So it would just keep on going, it would carry on. So you keep adding an alcohol, then a carboxylic acid, and so you end up with COO and OOC. So it's just reflected, that's all. But that's essentially the functional group for a polyester. So when we were drawing polymers yesterday, hopefully you remember we always do the square bracket and we write an N. So it's exactly the same with polyesters. So you wouldn't need to draw this, you would just draw that there, and then you would write an N. And obviously we have water released as well, so it's important that you remember the water. So this is AQA, condensation polymerization is all the exam boards, it's a higher tier topic, and it seems that all the exam boards want you to know is how to make a polyester, so a dioic acid and a diol, and then how to draw the functional group, which just looks like this. And as I said, you can just draw squares. Okay, so let's move on to today's topic then, if no one's got any questions. So today we are looking at kind of the biology of our chemistry or of our polymers. So yesterday we were looking at what we call synthetic polymers. So synthetic means kind of they were made, whereas natural polymers are ones that exist naturally. So they're in our bodies, so we don't need to make them. So the first natural polymer we're going to look at relates to carbohydrates. So sorry that you can't see all the screen there. So first of all, if you've covered this in biology already, you'll know that a carbohydrate is a compound that contains carbon, hydrogen, and oxygen. And that's really important because I've seen exam questions asking, what do carbohydrates contain? The way I remember it is the name carbohydrate almost has it in it. So carb, carbon, O, oxygen, and hydrate, hydrogen. So carbon, hydrogen, and oxygen. And this is the general formula. You don't need to know the general formula there, but I've just put it in there because some of the textbooks have it. So carbohydrates are split into simple carbohydrates and complex carbohydrates. So before we go a little bit further, I just want to remind you that when we make polymers, we always have a monomer making a polymer. So for example, in our condensation polymerization, we have our dioic acids and our diols forming our polymer, which is polyesters. And it's exactly the same with natural polymers. We have monomers that then form the polymer. So the third point on this slide talks about simple carbohydrates. So remember I said carbohydrates are split into simple and complex. So simple carbohydrates are called monosaccharides, and that is the monomer for complex carbohydrates, which are polymers. So I'm going to show you on the next couple of slides what the polymer of carbohydrates looks like. But essentially the monomer of the complex carbohydrates is monosaccharides. So that's the repeating unit that we need to know when it comes to carbohydrates. There will be a quiz at the end of the session, yes, and CX just means that that would be a number there. So usually there's a number there. Great. So for example, if you want me to fill in that for you, so glucose and fructose both have the same formula. It's C6H12O6. So actually X and Y there are the same. But what we can see is that if this is 6, that makes a C6. That would be 6. So H, 2 times 6 makes 12, and then O6. So glucose and fructose are both this formula here. But if you filled it, if you have other monosaccharides, you can try and make the formula there. But again, you don't need to know it. I've just put it in because some of the textbooks have it. The missing words, so simple carbohydrates are called monosaccharides. As I said, they are the monomers for complex carbohydrates. And these are sugars such as fructose and glucose. So fructose is on the screen, and here's also glucose. What we need to know about these, as I said, is that they are simple carbohydrates. They are monosaccharides. So that means they're also monomers. They're the simplest units that are repeated in polymers. Glucose does not have to be represented like this. You can just represent it as a hexagon. So just as a hexagon, that could be your glucose molecule. And fructose can just be represented as a pentagon. So they are slightly different to each other. Glucose is usually hexagonal and fructose is usually pentagonal, so 5 sides. They still have the same number of carbon atoms. So you can see the difference between glucose and fructose is glucose has one CH2OH group coming off, and there's a carbon at each point, or where the lines meet. So 1, 2, 3, 4, 5, 6. If we look at fructose, again, there's a carbon at each of the points. So 1, 2, 3, 4, and there's 2 coming off. So 5 and 6. So that's why fructose is pentagonal, whereas glucose is hexagonal. The second missing word is glucose. So it says simple carbohydrates are called monosaccharides and are sugars such as fructose and glucose. That's all it says. So they are sugars such as fructose and glucose. Okay, any questions about the monosaccharides, glucose and fructose? So remember, they're the monomers for complex carbohydrates. These are simple carbohydrates. We're going to look at complex carbohydrates in just a second. No? Okay. So this is not a complex carbohydrate. This is what we called a disaccharide. So remember, di means 2. So we just looked at monosaccharides, which are just one functional unit. So we've got the monosaccharides here on the screen. A disaccharide is when we join 2 monosaccharides together to form a molecule that's made of 2 monomers. Why are they different shapes? That's a really good question. So as I was just saying, it's to do with how they join up with their carbons and what their structure looks like. So fructose likes to have 2 carbons coming off of its structure, whereas glucose only likes to have 1. So that's why glucose is hexagonal and fructose is pentagonal. And as someone's just said, it's all to do with how what the sugars kind of taste like, as well as how healthy they are as well. So remember, their structures relate to that. So glucose is usually used for energy in our bodies. And doesn't fructose come from fruit? Yep, you can find fructose in fruit, yes. So it tends to be a healthier sugar than glucose. Good. So as I said, sucrose is a disaccharide. That means it's made from 2 monosaccharides. So on the screen, you can see 2 monosaccharides joining to form a disaccharide. And specifically in sucrose, it's glucose and fructose coming together to make sucrose. And the important point on this slide is the bond that's formed. So I don't know if you can see, but this bond here is a special bond. It's called a glycosidic linkage or a glycosidic bond, and that bond only occurs between sugars. So someone's just saying I've frozen. Have I frozen for anyone else, or am I not frozen? No, okay, that's great. So as I said, there's a glycosidic bond between monosaccharides of sugars. So specifically, we need to know about sucrose, which is a disaccharide. Di means 2 monosaccharide molecules come together to form a disaccharide. So as you can see, both these diagrams are showing sucrose. So that's really important. And the other important thing is that when they come together, this is a condensation reaction. And remember, condensation reaction means that water's released. So if we have a look, we can see that there's an OH group here, but what happens when they link is they just have an O bond, like so. So what's released is an OH plus a H, because only one of the O's is used, and that's water. So when this reaction takes place, this water is released to form that glycosidic bond. So this is a condensation reaction. It's not condensation polymerization, because we've not formed a polymer. But this is almost kind of a step down from that. It's still a condensation reaction where water's released, but it's not polymerization because we've just formed a disaccharide, which is just made of 2 monomers. Remember, polymers are made of lots and lots of repeating monomers. This is only 2, so it's not a polymerization reaction. Okay, any questions on sucrose? No? Okay. Okay, so someone's asking, what is a polymer? So yesterday we described polymers as molecules that have high relative formulae, so high relative formula masses. And we also said that as well as having a high relative formula mass, they are made of lots of repeating units. So lots of repeating units. Where does the O bond come from? So the O bond comes from these OH groups that are on the monosaccharides. So remember on glucose and fructose, if I just go back, you can see these OH groups. They have lots of these OH groups. And essentially when they try and join, these OH groups come together and you get an O bond formed between them and a water molecule comes out. So you get an O bond formed between them and a water molecule comes out. Okay, any question on that then? At all? Any questions on that? Someone's saying, for sucrose in the second image, why are there 2? Yeah, so that's a really good question. I couldn't find a diagram that showed sucrose and fructose joining up. So this is just an example. This isn't sucrose here. This is just 2 glucose molecules joining up, but I'm trying to show the glycosidic linkage there. So good question and good spot there. But no, that's not sucrose. This is sucrose. This is just showing what happens when you bind 2 monosaccharide molecules together. Okay, so hopefully there's no questions then on sucrose. If you have any questions as we're going along, please just pop them into the chat function. So now let's look at the polymers for carbohydrates. So we've had a look and we've seen disaccharides, we've seen the monosaccharides. The next thing we need to know about is the polymers. So is the water that comes out pure water? Yes, it is. It's pure water that comes out, H2O. Good. Okay. So starch and cellulose. So starch and cellulose are the polymers that form from monosaccharides and they are complex carbohydrates and they're called polysaccharides. So instead of being called polymers, we call them polysaccharides. So remember we had the monosaccharides, glucose and fructose, a disaccharide, which was sucrose, and now we are going to find out about polysaccharides or polymers. They are both made from glucose. So the monosaccharide in starch and cellulose is glucose, or the monomers are glucose. And they are formed from condensation polymerization reactions. So a water molecule is released and glycosidic bonds are formed. So remember I said that when we join up these molecules, we form glycosidic bonds. So the structure of starch is a lot less regular than cellulose, meaning that there are stronger intermolecular forces between the polymer chains in cellulose than starch. So I'm going to show you their structures in just a second, and hopefully you'll then be able to understand why there's stronger intermolecular forces between the polymer chains in cellulose than in starch. So I'm just going to answer some questions. I think we've just had some questions come through. Okay, that's fine. So starch is used as an energy store for plants, and cellulose is used in the cell walls of plants, giving them their strong structure. So does anyone know in biology how starch is used as an energy store for plants? Have you learnt that in biology, or why it's used as an energy store for plants? Can everyone still hear me? Tilly?
Hi, yeah, everything seems okay to me. Is everything okay on your end?
Yeah, is my video okay? It's not freezing?
No, it seems fine actually. I think occasionally your video, as in your face, is a little bit behind, but the screen and your sound is absolutely fine, so it's okay.
That's fine. Okay. Great. So we talked about starch being an energy store for plants, and cellulose being what's used in the cell walls of plants, giving them their strong structures. So what's really important is cellulose is used in the cell walls of all the different cells, and cellulose is quite a strong structure. And remember I said that it's got stronger intermolecular forces between the chains, so that makes it stronger than starch, whereas starch is used as an energy store. So saccharide just comes from carbohydrates. It's just a word that we use to describe the sugar molecules. So it's just a fancy word for sugar almost. Good. Okay, so let's have a look then at the structures of starch and cellulose. So good. Okay, so starch is on the left, and what you can see is that it's quite an irregular chain. So we've got lots of different branches, so it's quite irregular. So the intermolecular forces are not as strong as cellulose, where we've got these regular layers and this kind of regular chain. So that's why cellulose is a lot stronger. It's got stronger intermolecular forces between the polymer chains, whereas in starch, there's not those strong intermolecular forces. In fact, because it's so irregular, it's a lot weaker. And that's why cellulose is used in cell walls, whereas starch is not used in cell walls. It's used as an energy storage. Okay, good. So that's kind of all I needed to cover on carbohydrates. So the next thing we're going to look at is proteins, and proteins is a higher tier topic. So if anyone's got any questions on glucose or anything like that, then please send them in. If not, we'll move on to proteins. So I'll just see there are a few questions coming in. Bear with me 1 second while I have a look at these questions. Okay. Oops. Okay, sorry about that. You should be able to see my screen again now. Okay, so are there any other sugars apart from glucose and fructose? Yep, so sucrose is a sugar, it's a disaccharide. Some starch stores are in root vegetables, e.g. potatoes. Yep, that's a really good point. And starch, as I said, is used as an energy store in plants. Good. So we don't tend to store starch, but starch is used in plants as an energy store. Excellent. Okay, good questions, everyone. So let's move on to proteins then. As I said, a higher tier topic. So proteins are also condensation polymers, which are formed from amino acid monomers. So remember, in carbohydrates, the monomers are the monosaccharides, which are glucose and fructose, mainly glucose, whereas in proteins, they are formed from amino acids. And these are joined together by what we call a peptide bond. So remember that we talked about glycosidic bonds with glucose and with carbohydrates. In proteins, we have peptide bonds or peptide linkages. And these are formed when a carboxylic acid and an amine react. So on the amino acid structure, which you can see on the screen, one end has an amine group, which is basically comes from ammonia. So ammonia is NH3, so we've got NH2 there, so it's an amine group. And the other end is our carboxylic acid group, which we know lots about. The R chain is just a side chain, so lots of amino acids have different R chains. The simplest amino acid is glycine, which just has a H here. So this could be anything. There are about 20 amino acids, and there's lots of different R chains that they could have. So the main thing that we need to know then is that these amino acids join together to form an amine or a peptide link. And this peptide link is when the carboxylic acid group joins to the amino group. So on the next slide, we're going to see what that linkage looks like. So let's have a look. Okay, so here you go. So I've joined here, or the picture's joined up 3 amino acids. And as I said, it's the simplest amino acids that are being joined here. And that is glycine. So instead of this being an R, this says H. So the simplest amino acid just has a hydrogen. And what's happening is when the amino acids join, remember they've got the amine group on one end and the carboxylic acid group on the other end. So when another amino acid comes along, we end up forming this here. And this linkage here is called a peptide bond. So it's the bond here between the CO and the NH. So just here. Should be able to see that now. So just on the screen, the CO and the NH, that is what we call a peptide bond. And you can see that there are 2 of these peptide bonds on the screen. So there's one here and one here. And they're really important. So we had glycosidic linkages with carbohydrates where it was just oxygen in the bond, but with amino acids, we've got the C double bond O and the NH group. And that is what forms the peptide bond in proteins. So this is a tripeptide, this is not a polymer. So remember, polymers are made of lots and lots of amino acids, whereas here this is just made of 3, so it's not a polymer yet. It's a tripeptide because it's made of 3 glycine molecules. Great. Okay, so let's have a look then at the next slide, which is DNA. So that was proteins done. The last thing we need to cover is DNA. Someone's asking, in higher GCSE, do we need to know all of the chemical formulae? So you need to know everything that is in the textbook, essentially, for whichever exam board you have. So all the quantitative chemistry, you should know all the equations for. You do need to know kind of all the equations for your organic chemistry. So all the 4, so remember we learned 4 from each functional group. So for example, the first 4 alcohols, the first 4 carboxylic acids. So it's important that you know that. Do we need to know the formula of peptide? So not peptide, you just need to know what the bond is. So the specific peptide bond is CO, so it's a C double bond O NH, and you need to know that bond, or you just need to be able to name it. So in an exam question, they might ask you, what are the bonds between amino acids? And you need to say it's a peptide bond. That's what it's called. Okay, so now let's have a look at the third kind of natural polymer, which is DNA. So DNA makes up all of our cells. It's in the nucleus, which I'm sure you're aware of if you study biology. And again, it's a polymer. It's made of condensation and polymerization, but the repeating units in DNA are called nucleotides. So remember, in carbohydrates, the repeating units were monosaccharides. In proteins, the repeating units were amino acids. And in DNA, the repeating units are nucleotides. So DNA is known as a polynucleotide, which is just another word for a polymer, but specifically a polynucleotide. So remember when we looked at carbohydrates, the word for their polymer was polysaccharide, and the word for the polymer for proteins is just protein. Or you can say polyamines, but I prefer to say proteins as the polymer there. So what we need to know then is what happens when we join our nucleotides. So when we join them, we get DNA and water, because again, it's a condensation polymerization reaction. So DNA and water is released. And all of these natural polymers are condensation polymerization reactions, so water is always released. There are 4 nucleotides. So when we were looking at carbohydrates, we saw that there were 2 monosaccharides, which were glucose and fructose. Amino acids, there's lots of them. You don't need to know about all of them. You just need to know that there's a peptide bond between amino acids. But in DNA, we do need to know that there are 4 nucleotides because there are only 4. Will this be on YouTube, as the past chemistry webinars weren't on YouTube? I'm not sure why they weren't there. They should be coming up soon. I don't know. Tilly, do you know why they're not up yet?
Which ones do you specifically mean? Because all the ones from this week so far are up there. Let me know the specific one that you can't find, and I'll let you know if it's up there or not.
Okay, and someone's saying, if you decide to pick chemistry, do you have to have some background biology knowledge? Do you mean pick chemistry at A level? If you do, no. Chemistry A level's not that biological. This is really the only kind of biology-based topic in chemistry GCSE. But no, you don't need to. I would pick chemistry A level if you enjoy chemistry. That's what I said last week as well. Okay, great. Anyone that's sending me questions on the chat function, I can't currently open the chat box. So if you send them in the Q&A instead, I can open them there. So for some reason, it's not letting me open the chat box. So if you have any questions in the chat box, just send them onto the Q&A function instead, and I'll answer them there. Okay, so the third one then is DNA. So as I said, condensation polymerization. So it releases water when they add together or when these nucleotides join together. And the 4 nucleotides that we need to know about are adenine, thymine, cytosine, and guanine. Now, I appreciate that these names are a bit difficult to remember. So it looks like at GCSE, you don't really need to know the names, but you do need to know the letters and specifically which nucleotides they bond to. So there's a specific rule, and that is with DNA, we can't just bind adenine to cytosine and cytosine to thymine. They have to bond in their pairs. So adenine will always bind to thymine, and cytosine will always bind to guanine. So I'm going to show you a structure in just a second, and you'll see that. But it's really important that you won't have any mixed pairings there. They will always bind in those pairs. So when the nucleotides come together to form their polymers, they have to be in that order. Have to be adenine to thymine and cytosine to guanine. Great. Someone's asking, how many A levels did you do? I did 4. So I did biology, chemistry, maths, and Latin. But I think it's more usual to do 3 these days. I don't think many people do 4 anymore. Okay, so any questions on nucleotides before we look at the structure of DNA? As I said, if you can send them into the Q&A function instead, that would be great, just because I can't see the chat box at the moment. Is it possible to do 5 A levels? Yep, it's definitely possible to do 5 A levels. If you think you're up to the challenge, then yeah, definitely do it. I have a lot of friends that did 5 A levels, and what I would say is it's very difficult. You don't get many kind of free lesson time. So one of the main things at A level is that often you get what we call free periods or free lesson times, and that's where you kind of catch up on work and revise. So if you don't have many of those, it can be quite difficult. So I didn't have many, and I did 4, and a lot of my friends did 5 and they had none. So I'd just be kind of wary on how much time you think you need to study for your A levels. Okay, is this in the baccalaureate scheme? So IB, I'm not sure what you're talking about. So this is GCSE. Are you talking about the IB equivalent to A level? I'm not sure. If you could rephrase your question, that would be great. Okay, so let's look at the structure of DNA. So hopefully the next, just bear with me. Okay, should be coming up in just a second. There we go. Okay, so this is our DNA structure. And first thing I want you to look at is this screen or this picture here. And what we can see straight away is that these nucleotides, C is bound to G, A is bound to T, G is bound to C, and T is bound to A. So remember, that is the order that it happens in. They will not bind in any other way. They have to bind in those pairs. And what we need to know about DNA is that it's a double helix. What that means is that it's essentially 2 polymer strands. So this is one polymer strand, this is another polymer strand. and they're binded together. And where they are bound together, we get this kind of twisting function, which makes it a helix. So it's one of the only structures that forms a double helix. And as I said, the important thing is that C binds to G, A binds to T. There's 2 polymer strands, and we form a double helix. The bonding between the nucleotides is known as hydrogen bonding. So it's hydrogen bonding. You don't need to know any more than that. You just need to know it's hydrogen bonding. So we've looked at carbohydrates, and carbohydrates formed glycosidic bonds, remember, with the O. With proteins, we formed peptide bonds. So that was the C double bond ONH group. And with DNA, we form what's known as a hydrogen bond. So it's specific that you need to know just the bonding between them. Okay, what is the maximum number of A levels you can do? I'm pretty sure it's 5, but most schools don't like you doing 5 anymore. I think most schools now advocate doing 3. The maximum you can do at GCSE, so I did 12 GCSEs. I think you can do up to 14. Again, it's up to what your school allows. There's no kind of maximum cutoff. Someone said I cut out. Can you explain that again? Yep, so I'll explain the DNA again. So DNA is a double helix, which means it's formed from 2 polymer strands that are twisted together. So if you have a look at this structure here, you can see that twisting kind of structure. In DNA, you always have the same nucleotides joined together, so C to G and A to T. And what's really important is the bonding between those nucleotides is hydrogen bonds. So nothing else, just hydrogen bonds. Would it do the same thing if you flip the DNA double helix? I'm not sure what you mean by flip the DNA helix, but yeah, we can have this polymer on the left, for example, could be on the right, and that one could be on the left. The important thing is it would do that as long as you have your nucleotides joined up in the correct way. So it has to be C to G and A to T. Okay, so that's kind of all the content that I wanted to cover today. So if we go back to our learning objectives from yesterday and today and see if we've been able to cover it all. So yesterday we looked at drawing the diagrams for addition polymerization, which we managed to do. We looked at knowing the uses of some of the polymers. Again, we looked at the 4 most common polymers. So we looked at polythene, PVC, polytetrafluoroethylene, and polypropene. We looked at the disadvantages of polymers. So remember, they're non-biodegradable. They use up landfill sites. It's difficult to recycle them because they require a lot of energy. We have to separate them. And higher tier was to be able to draw the diagrams for condensation polymerization, which, as I said, we weren't able to go over yesterday, but we did that today at the beginning of the session. So hopefully that's now something that we're aware of. Does anyone have any questions from yesterday's session or anything they want me to go over again from yesterday's session at all? How many GCSEs can you do if you're homeschooled? I'm not sure about that question, sorry. I'm not sure. I think, again, it's completely up to you however many you want to do. There's no kind of cutoff. I know you definitely have to do the core subjects, so maths, English, science, but I don't think there's a cutoff as such. Okay, so there don't seem to be any questions about what we covered yesterday. So let's look at what we covered today. So we need to know how sugar molecules polymerize. So we looked at that. So we had our monosaccharides, which were glucose and fructose. They bind together to form glycosidic bonds where a water molecule is released to form polymers, and their polymers are known as polysaccharides. And the 2 polysaccharides we need to know about are starch and cellulose. Cellulose is stronger than starch because it has stronger intermolecular forces between its structure. So remember, cellulose's structure is more regular, it's nice layers, so they have stronger intermolecular forces, whereas in starch, it's more of an irregular chain, so there aren't that strong intermolecular forces. As I said, they come together and they form glycosidic bonds between the sugar molecules or between the monosaccharides. So that's how sugar molecules polymerize. Amino acids are the building blocks or the monomers for proteins, and they come together to form a peptide bond. So lots of amino acids come together to form a peptide bond, and the polymer for amino acids is known as proteins. And then finally, we looked at the structure of DNA. So DNA, the monomers are nucleotides. And there are 4 nucleotides, A, T, C, and G, adenine, thymine, adenine, thymine, guanine, and cytosine. Uracil is another nucleotide, but it's not present in DNA. So if you do biology, you might know uracil is present in RNA, which is a different type of molecule. We don't have RNA in our cells, we only have DNA. So does anyone know what does DNA stand for? If you could put it in the Q&A function, that would be great. What does DNA stand for? Someone's asked me to go back to the double helix slide. I will go back to that in just a second. Did you have to do core PE at GCSE? No, I did not do PE at GCSE. Good, deoxyribonucleic acid. Excellent, well done everyone that's answered that. So deoxyribonucleic acid. Okay, so that's what DNA stands for. Again, you just need to know the 4 nucleotides, and remember that A binds to T and C binds to G, and they come together to form a hydrogen bond. If you're sending me questions in the chat box function, can you send them in the Q&A instead, please? Good. So someone's asking me, can you explain about sugars again? Yep, sure. So let me go back to the slide on sugars and see if we can explain that again. Okay, so let's go to this one here. So sugars are made from monosaccharides, which are glucose and fructose. They are the monomers, and they come together to form a glycosidic bond. So I'll show you the glycosidic bond on this slide. So this is the glycosidic bond here. So glucose and fructose come together to form this glycosidic bond. And what's released is water. So H2O comes out. And that's what makes it a condensation reaction. So the polymers of sugars occur when lots of glucose molecules join up together. And the 2 polymers that we need to know about are starch and cellulose. So you can see those on the screen. So starch has this irregular structure, whereas cellulose has this much more regular layered structure. And that means that the intermolecular forces between these layers are essentially stronger in cellulose than in starch. And that's why cellulose tends to be used in the cell walls of plants to help them keep their structure, and it's stronger, whereas starch is used as an energy store. So it's not really that strong, it's a little bit more flimsy, so it's not used as a structural sugar. Okay, so someone asked me to go back to the structure of DNA, so I will do that now. So this was the structure of DNA. So again, you can see the nucleotides join up together. So C binds to G, A binds to T, and it's a hydrogen bond between them. There's 2 layers of polymers, or 2 chains of polymers, which bind together and form this double helix. Okay, so I think that's kind of a recap of everything we've covered today and yesterday. We will now do a quiz. Can you go over the first slide? Okay, fine. Let's do the quiz, and then I will go back over the first slide and amino acids again. Okay, so if you go on to menti.com and type in this code 202397, hopefully you should be able to join the quiz. So 202397. Great, so I can see lots of you joining. Go to menti.com. Someone asked me to go over the first slide again. I will go over that, and I will go over amino acids after the quiz. Good, so I've got about 14 of you in the quiz at the moment. I'll just wait for a couple more to join. So I think there are 40 of you in the webinar, so I'll just wait for a couple more. Someone's asking for the link. You should just be able to go to www.menti.com and type in that code. Okay, I'll start it in about 10, 15 seconds. Okay, let's start. So the first question should be coming up on the screen in just a second. Which of these is a monosaccharide? Which of these is a monosaccharide? So monosaccharide, remember that's basically the monomer. So the answers on the screen are sucrose, starch, cellulose, and glucose. Which of them is a monosaccharide? Okay, and the answer is glucose. So sucrose is a disaccharide. It's formed from 2 molecules of glucose, well, 1 of glucose and 1 of fructose. Starch and cellulose are polysaccharides or polymers. They're formed from lots of monomers of glucose. So glucose is the monosaccharide. So well done everyone that got that correct. Excellent. Okay, so the next question should be coming up in just a second. Which bond is formed between glucose monomers? So which bond is formed between glucose monomers? Is it a peptide bond, a glycosidic bond, a hydrogen bond, or is it intermolecular forces? What do we think? Which bond is formed between glucose monomers? Okay, yep, so the answer is glycosidic linkage, which most of you got, which is great. So peptide bond is formed between amino acids. Hydrogen bonds are formed between nucleotides. So nucleotides are the monomers of DNA, and intermolecular forces are formed between chains of polymers. So not in carbohydrates, we get glycosidic linkages between carbohydrate monosaccharides. Well done, everyone. Okay, so next question should be coming up on the screen in just a second. So amino acids contain which groups? Amino acids contain which groups? So remember when we looked at the structure of amino acid, they had one group on one end and one on the other end. So was it ester and oxygen, amine and ester, amine and carboxylic acid, or amine and alcohol? What do we think? Good, so the answer is amine and carboxylic acid. So well done, most of you got that, really well done. So ester and oxygen doesn't really exist, amine and ester again doesn't exist, and amine and alcohol, not really. So it's always amine and carboxylic acid. So they'll have the NH2 on one end, which is the amine group, and the C double bond O, OH group on the other end, which is the carboxylic acid. Great, well done. So the next question should be coming up. What are the monomers that make up DNA called? So I think you have to type in the answer to this one. So what are the monomers that make up DNA called? So we actually went over this just a few questions ago. So it's one of the answers to a different question. So what are the monomers that make up DNA called? Okay, so the answer is nucleotides. So if we have a look, there we go. So the correct answer is nucleotides. Good, well done everyone. So yep, CGAT, I would give you that as well. So it's spelt nucleotides. AT, C and G are all nucleotides. So you need to know the actual name, which is nucleotides. Great. So hopefully the next question should be coming up on the screen. Adenine binds to which nucleotide in DNA? So adenine is a nucleotide. Which nucleotide does it bind to? So remember there are pairings. They have to bind to their specific pairing. So is it thymine, guanine, cytosine, or uracil? What do we think there? So the correct answer is thymine. So adenine binds to thymine. In RNA, if you've studied it, it binds to uracil, but in DNA, which is what's in the human body, it binds to thymine, which pretty much all of you got right. So very well done. Okay, so hopefully now it's going to show the leaderboard, just coming up on the screen. So well done, everyone. You all did very, very well. So well done, Lala, you are our winner today. Excellent. Okay, so if I now go back to the slides. So someone asked me to go over the first slide again. I think that was condensation polymerization. So hopefully you can see on the screen what I drew for condensation polymerization. So I drew a diol, which is a dialcohol, and a dicarboxylic acid. And these essentially join together to form an ester bond, which is COO. And when they join together, they form a polymer or a polyester. So the ethane diol and the and hexane dioic acid join together to form a polyester. And what you need to know is that it's a polyester bond that's formed, and we use a diol and a dioic acid to do that. So that just means there's two of the functional groups on either end. So if we look at the carboxylic acid I've drawn, you've got the COOOH on one end and COOOH on the other end. With the diol, again, you've got the OH on one end and an OH on the other end as well. Okay, so let's try and do some exam questions then. What I'm going to do, if anyone's got any questions, just pop them into the Q&A function and I'll try and answer them. Yep, so someone's asking, can you please go over the fourth slide? Okay, I'm not sure what the fourth slide was, but yes, I think it was carbohydrates. Okay, so we've got sucrose there. Is this the slide that you wanted me to go over? So the person that asked me to go over the fourth slide, was it this one? Okay, so that person is saying yes. So ethane diol is just two carbons, hydrogens, and two alcohol groups. So that's what ethane diol is. So this is the fourth slide. It's sucrose. It's a disaccharide, which means it's formed from two monosaccharides. Sucrose is specifically formed from one glucose and one fructose, making this glycosidic bond here. And that glycosidic bond comes from these OH groups. The OH groups join together to form an O bond, which is a glycosidic bond. And then that means that two hydrogens and one oxygen come out in the form of water. So sucrose is a disaccharide, two monosaccharides come together. Polymers or polysaccharides are formed from lots and lots of different glucose molecules. Amino acids, yeah, let's go over that. So proteins are formed from amino acids, and amino acids have an amine group on one end, which is NH2, and a carboxylic acid group on the other end. And when amino acids join together, the carboxylic acid group binds to an amine group of another amino acid. And we form what's called a peptide bond. And you can see the peptide bond on this slide here. And what you can see is this double bond, C double bond O, and this NH. And that is what we call a peptide bond. And there's two here because we've got three amino acids joining together to form these peptide bonds. So what you can see is this is one amino acid. It had its amine group on one end, and this would have been a carboxylic acid group. But what's happening is it's binding to the amine group of another amino acid. And so it's lost its OH group, and this has lost one hydrogen, which is OH, and hydrogen makes water. So that's why it's condensation polymerization, because it's losing the water. Good. So I'm keen to do some exam questions. Someone's asking, how many years have I been teaching? I think 3 or 4. I'm not entirely sure the answer to that question. Okay, so let's do some exam questions then. Actually, it's not that slide. Okay, what I'm going to do is, okay, just bear with me. I'm just going to share the questions instead. Okay, hopefully you should be able to see in just a second the first question come up on the screen. There we go. So polymers are addition or condensation polymers, and polymers can be formed by using monomers. Explain, using appropriate monomers from figure 12, how different polymers can be formed. So we need to use the monomers in that table to explain how polymers can be formed. So take a few moments to think about that and tell me what you think. Someone's asking, is chemistry the only science you took at A-level? Nope, I took biology, chemistry, Latin, and maths. How many lessons have you taught? I've taught all the chemistry online school lessons, and I've taught about 240 on MyTutor. So they're the private tutoring on MyTutor, so the one-to-one tutoring. Okay, so anyone got any answers to this question then? So using the table on the screen, how different polymers can be made. What do we think? As I said, if you can answer in the Q&A instead, that would be great. Good. Okay, so someone's given me part of an answer. They've said chloroethene by breaking that double bond. Yep, so the first polymer we can make on this screen is addition polymerization. And remember, addition polymerization is when the monomers come together to form a polymer and nothing else is produced, just the polymer. So chloroethene is what we looked at yesterday. It forms PVC, or polychloroethene, when the monomers join up. And essentially that double bond here that we can see between the CCs break. And when they break, we can form polymers, which is known as polychloroethene. And as I said, that's addition polymerization. What about these two? What do they form? Ethane diol and ethane dioic acid. I covered that today. What's it called? Why does it happen? So if you're going to answer that question, if you could put it in the Q&A, that would be great. So what's formed when ethane diol and ethane dioic acid react together? Good, it's a condensation reaction, excellent, and you get water as a byproduct. So water as a byproduct. So they form a polyester. So you don't need to know what the name of the polyester is, you just need to know that it forms a polyester, and it's a condensation reaction where water is removed. Excellent, any questions on that at all? So well done to people that answered that. That was really good. Yep, okay, fine. Let's move on to the next question. Oops. Okay. All right, so the next question isn't showing up. Let's see if I can get this one to show up. No, okay. All right, so I think we'll end the lesson there then. Sorry about that. We'll try and do some more exam questions tomorrow morning before we start Intro to Electrolysis, but for some reason the lesson space doesn't want to show the other exam questions. So we'll finish there. Thank you everyone for joining me today. As I said, we'll be moving on to electrolysis tomorrow. So we're done with polymers, and hopefully this lesson will be up soon on YouTube. All right, take care everyone. Bye.